Hello students, welcome to LEELA'S tutorial. So today we will discuss about somaclonal variations. So why these variations occur, right? And what is the reason and whether this character is a beneficial or a harmful process. Okay, so first the somaclonal name itself implies that the cells or the plantlets are produced from the somatic cells. Right? So here in plant tissue culture, we produce diploid plantlets and also haploid plantlets. If we are producing the diploid plantlets, they are produced from the somatic cells of the parent plant. If it is a haploid, they are produced from the gametic cells of the parent plant. Is that clear? So here in these variations, we are talking about the cells or the somatic cells which are taken as the explant. Okay, so either in case of clonal propagation or the micropropagation, first you are going to have an intermittent stage, that is the mass of cells called as callus. Right? The callus is a mass of undifferentiated cells. Right? These are unorganized cells which later transform into or develop into the shoot and root system. Right? So here when you are getting that clone of plantlets, during this plant tissue culture, there may be certain variations or the changes that may occur. Right? So these changes are referred as somaclonal variations, and the plantlets which are produced by this process are referred as somaclonal variants. Okay? So let us see here. This is the parent plant. And from the parent plant, I can take any part of the parent plant, the somatic cell, right, or the somatic tissue. So here I have selected the leaf. So I am excising the required size of the explant and inoculating in plant tissue culture media. So once I inoculate, I incubate them under favorable conditions. So here auxin to cytokinin ratio, that is the growth hormone ratio, must be 1:1, right? So auxin to cytokinin ratio, when it is 1:1, it will result in formation of callus, right? So if I want the shoot system, what I will do? I will increase the cytokinin and decrease the auxin. So I can take 1:3 for shoot formation, and again if I want the root, I can increase the auxin and decrease the cytokinin. As a result, I can get the plantlets. Right? So this is called as your propagation, or clone of plantlets are formed from a mass of tissue called callus. Is that clear? First you are selecting the parent plant, disease-free plant, then the explant, excising the required size of the explant and inoculating in the media. Then you are allowing it to form a callus, and then from the callus you are getting the plantlets. See here in the plantlets, you can see in the progeny, you can see there are variations in the plantlets which are produced. So in the first case, the length of the plant is very much high, and in second case you can see the pigmentation. The dark pigmentation is observed in the leaves. Then in next one you can see the spotted, right? The spotted pigments are seen, and in last one it is similar to the parent plant. Is it clear? So that means there are certain variations out of hundreds of plants which are produced by this micropropagation process. During this process, there are certain plants which are showing some changes. These plants which are showing these changes are referred as somaclonal variants. Okay, so here I have shown the variation in these three plants. Right? So as they are visible to our eyes, that means the external morphology is different from the parent plant. So these variations are referred as phenotypic variations. Right? So here it is resembling the parent phenotypically. That means external morphology is same as the parent plant, but we don't know whether the change is genetically also. Right? So for the genotypic changes which cannot be visualized by simply checking the external morphology, for that you have to go for your molecular markers. Okay, then you can say whether it is a variant or the normal identical parent plant. Okay, so let us go into detail. Here, the changes in regenerated plants are occurred during this plant tissue culture. Okay, so here they are not identical to the parent plant. So as I have shown, there may be change in the leaf structure, change in the fruit color, or change in the flower color, or pigmentation, height of the plant, or it can be some other characters. Okay, so it will not resemble to the parent plant. And these somaclonal variations are classified into two types based on their inheritance, right? Whether they are these variations or the changes which has been occurred, whether they are going to transmit it to the next generations or not, right? If you are sowing these seeds, right, you are getting the seeds and you are sowing these seeds, if you get the same type of plant, then you call it as heritable variation. After sowing the seed of this plant, if you are getting the normal parent plant, then you call it as non-heritable variation. That means the variation is only to that progeny. It is not inheriting or transmitting it to the further generations. Is that clear? So based on that, the two types of somaclonal variations are heritable variations and non-heritable variations. The heritable variations are referred as genetic variations and non-heritable variations are referred as epigenetic variations. Okay, so again the genetic variations are classified into two types where you can see the pre-existing changes in the somatic cell. If there is a pre-existing change in the somatic cell, that will not be inherited to the, that will be herited to the progeny, right? Because the change has been occurred in the somatic cell. Then other thing is due to the mutations. If the mutation in the DNA is high, that means the frequency of the mutation in the DNA is high, means again that cannot be reverted and it will be inherited to the next generations. Okay, first one is if the variation is pre-existing in the somatic cell or if the mutation is occurring at high frequency, they will be inherited. That variation will not be reverted back. Is that clear? Next, coming to the epigenetic variation. So these epigenetic variations are non-heritable. That means the changes which have occurred in the plants will remain only to them. They are not inherited. These changes are not inherited to the next generations. Okay. So here, why these variations have occurred? This may be due to the plant tissue cultural conditions. It can be due to the media components or the external environmental conditions. And the second one is the temporary phenotypic changes. So temporary phenotypic changes may occur which may revert back after attaining the favorable conditions. Then the last one is decreased frequency of DNA mutations. So if it is a very low DNA mutation, that can be corrected, right, in DNA repair and all. So here these are the two types because of which the variations are occurred. Okay, so it can be a heritable or non-heritable. So what is the reason why these heritable and non-heritable variations are occurring? Let us see. Again, the reasons or the cause for these variations is classified into three types. The first one is physiological cause, second one genetic cause, and third one is biochemical cause. So in case of first one, the physiological cause, it is mainly due to the tissue culture conditions. The plant tissue culture, the growth hormones what you are providing. Plant growth hormones, right, such as auxin, cytokinin, gibberellins. Okay, so all these hormones will affect and cause some variations in the plants, right? Not only the media components but also the cultural conditions such as pH, temperature, light intensity, right? Antibiotic presence or addition of more amount of antibiotics, right? So all these. And the second one is the genetic cause, right? So the genetic cause may be due to the structural aberrations or numerical aberrations, right? The structural aberrations means there is structural change in the chromosomes. Okay, so when the structural change will be there, when there is any addition or deletion. Otherwise, you can say in case of structural, the structure of the chromosome is changing. That means if you have any genes A, B, C, there may be B, A, C. That means their arrangement is changing, right? But the number of genes present are same. Is that clear? So only there is a change in the structure but not in the number, right? This change in the structure may be due to base addition, deletion, translocation, transversion, inversion, right? So the structure of the chromosome may change. In other case, it is a numerical aberration where the number of chromosomes may increase or decrease, right? So that is the aneuploidy condition. So in aneuploidy condition, there may be plus or minus one chromosome. There may be one chromosome more or there may be one chromosome less, right? Otherwise, it can be the polyploidy condition. So in case of polyploidy condition, either there may be addition of complete set of genome, not single chromosome. Complete set of genome may be increased or decreased, right? So like that, here the aneuploidy or polyploidy where it is a beneficial character in plants. When you take that in case of humans or animals, these decrease or variations in the structure of the chromosomes or the number of the chromosomes may result in the syndromes, right? So Turner syndromes and all, right? Whereas here in plants, it is a beneficial character where you can produce the plant with different colored flowers and other beneficial characters. So let us see that in applications, right? The third reason is biochemical cause. In case of biochemical cause, the components whatever you are adding in the media, they undergo certain biochemical reactions, right? In case if you are growing the plants in dark, the light intensity is not proper, the lack of sunlight, then in that case there may not be any photosynthesis. Okay, in some other case there may be lack of nitrogen metabolism or production of starch. They may enter into the carotenoid pathway. So like that, there may be some biochemical changes which may induce certain variations in these plants. Is that clear? So these are the three reasons why the plants are showing those variations. So how you can know at the younger stage itself, when you are shifting after micropropagation, you will be shifting them to the greenhouse, right? So in the greenhouse, when you are separating them, separating all the clone of plantlets, you can go for your molecular marker screening. So in this screening, by using the molecular markers, you can check for the phenotypic variations and the genotypic variations. Phenotypic variations which may seen by the viewing the external morphology of the plant, that is color of the flower, color of the fruit, right, leaf structure and all. If it is a genotypic variation, that is it can be within the genes. Okay, so the genotypic variations may cause some antibiotic resistance, okay, increased yield, or it can be tolerant to some other toxins, like that. Which cannot be visualized. It is not the external, it is the internally variation which is occurring inside the genome. Okay, so here after this, let us see the applications. As I told, the somaclonal variations are the beneficial characters in the plant biotechnology, so that we can produce a new variety of plant, okay, and also we can include some new characters, and unexpectedly you may get a new trait or new variety of plant. Is that clear? So the first application, by this technique or by having the somaclonal variations, these somaclonal variants or the plants may show biotic and abiotic stress tolerant. Right? Biotic means with some other living things. Abiotic, the non-living things like light, temperature, pH, etc. Right? So they are tolerant to the biotic and the abiotic stress, and then they may increase the yield of the crop in the agricultural fields. Then it may be a crop improvement where you can produce a fruits of bigger size, or you can produce a flowers of different colors on single plant. Then it is increased secondary metabolites. So by these variations or minor mutations, you can produce your secondary metabolite in bulk amount. Okay, and also you can avoid the intermittents, or you can avoid certain interrupting biochemical pathways. Okay, so like that you can increase the production of your secondary metabolites. Then you can grow the plants in high pH or low pH and in increased salt conditions, that is the salinity. Right? Even in nowadays they are growing the plants using ocean water also. That means they are salt tolerant plants. Then you have antibiotic resistant plants. Then drought resistant, where the plants can grow without any water or with minimum amount of water, where you can see the drip irrigation, only one drop of water is required for them. So like that the drought resistant plants can be produced, disease resistant plants can be produced, and these plants may be tolerant to the toxins, or they can be resistant to toxins, herbicides, etc. Is that clear? So here the somaclonal variants are the plants which are beneficial to the plant tissue culture. And if you have any doubts, you can put it in comments, and subscribe for further videos.